Okay, the Krebs cycle, or it's also called the citric acid cycle, the exact same thing, just different names. Right where we left off was the transition step, and so we have acetyl-CoA now, and so that is going to kickstart this process. The goal of the Krebs cycle is to produce high-energy electron carriers to be sent to the electron transport chain. So that's NADH, FADH2. Those can be sent to the electron transfer chain to go ahead and make ATP energy from that. And we'll go over the electron transport chain later. But we're starting with acetyl-CoA. Now, if you remember, this oxaloacetate from gluconeogenesis seemed kind of random because it was the only one with an intermediate step to go back up. But now it's going to start to make a little bit more sense because you need acetyl-CoA plus oxaloacetate for the first step of the Krebs cycle. And I'm just going to make a note here that this is from gluconeogenesis. So the first step is taking these two and it's turning it into citrate plus CoA-SH. So citrate and CoA-SH. And this is done by an enzyme called citrate synthase. That one is fairly easy to remember since we are synthesizing a citrate. And if we're keeping track of our carbons, we determined, if you remember from the transition step, that acetyl-CoA is 2 carbons because we start off with this 3-carbon pyruvate. We lose 1 carbon going through the transition step to make acetyl-CoA, which has 2. Now for oxaloacetate, if you remember, pyruvate goes to oxaloacetate, but it's a carboxylase, which means it's going to be adding on a carbon. So oxaloacetate is actually going to have 4 carbons. So we're adding a 2-carbon molecule plus a 4-carbon molecule. So citrate plus CoA-SH will have 6 carbons. From here, we are forming isocitrate. And the enzyme that catalyzes this is called aconitase. And if you could tell by the names already, this is just an isomerization, fairly simple step. And from isocitrate, we are going to form alpha-ketoglutarate. And the enzyme that catalyzes this is called isocitrate dehydrogenase. Alright, and this step is also important because it forms NADH. And so this is one of those high energy electron carriers that we were talking about that is going to move to the electron transport chain to make a lot of ATP. This step also releases a CO2. So we lose a carbon here. Now, if we're keeping track of our carbons, I'll go ahead and make note of all the carbons. So we said acetyl-CoA had 2 carbons. Oxaloacetate, we said 4 because we added on 1 from pyruvate. And we said that this here, citrate, had 6 carbons because we were adding those 2. So now this is just an isomerization reaction. We're not gaining or losing any carbons. So this is also going to have 6 carbons. And then now we're losing a CO2 to go from isocitrate to alpha-ketoglutarate. So this is going to have 5 carbons. Next, we move on to form succinyl-CoA. And the enzyme that catalyzes this is alpha-ketoglutarate dehydrogenase. Now this step is very similar to the step above it. We're going to be also reducing an NAD to form NADH, and we're also going to lose another CO2. So that means that succinyl-CoA will have 4 carbons. And if you notice, the enzymes are named similarly, isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase. So those 2 steps can be seen as kind of like twins. They're very similar, just the names are a little bit different in the number of carbons. Next, we are moving on, and from here we are forming succinate plus CoA-SH. And this is done by an enzyme called succinyl-CoA synthase. And in this reaction, we are not losing any carbons, but we are forming a GTP, which is another high energy molecule. GDP can ultimately get turned into ADP as well. From here, we are forming fumarate, and this is done by an enzyme called succinate dehydrogenase. And so it's a dehydrogenase enzyme, so we can kind of assume that we're going to be doing some kind of oxidation and reduction. So this one in specifically is a little bit different because we are going to be reducing FAD to form FADH. This is also one of those high energy molecules that is going to be sent to the electron transport chain to contribute to the proton gradient. Next, from here, we are going to form L-malate. And the enzyme that forms this is called fumarase. And this is actually a hydration reaction. Nothing else happens in this step, so we're going to move right along to form back to oxaloacetate. Now, this might look familiar because we've seen oxaloacetate quite a bit now. The enzyme that catalyzes this reaction is called malate dehydrogenase. Not gaining or losing any carbons, but again, it is a dehydrogenase, so we're going to be doing some kind of oxidation and reduction. This time we're going back to just forming NADH. And if we're counting our carbons, it should all make sense because we left off with 4 carbons up here. And all the way down, we did not gain or lose any carbons. So 4 carbons and everything should check out. If you come up here, we said that oxaloacetate had 4 carbons, so everything should be balanced. Now, if we kind of want to get a bigger picture glimpse of what's going to go on after this, we can show that from here, NADH can go through oxidative phosphorylation, which is going to form ATP and NAD. And we're also going to need O2 plus ADP. So as you can see, NADH, this highly reduced molecule, ended up producing ATP. So that's kind of the goal of this. If we scoot back up here to when we made a GDP, if we use an enzyme called nucleoside phosphate kinase and also an ADP, we can form also ATP plus GDP. So we turn that GTP into an ATP, which is more useful for us. So we can zoom all the way out and kind of see what we're working with. We started from acetyl-CoA and we went all the way down back to oxaloacetate. And in the pathway, in doing so, we created a lot of high energy, highly reduced molecules. NADH, FADH, all of those can be sent to the electron transport chain to make ATP. And we did make 1 GTP, which can also be turned into ADP. So again, the goal, obviously in metabolism, is to produce ATP, to produce energy. So that is it for the Krebs cycle. All of the notes from this will always be in the link tree, biochemistry, Krebs cycle / citric acid cycle. It's the same thing. Next time I'm going to kind of talk about the breakdown of fatty acids and how oxaloacetate plays into all of that. It's not a straight down pathway like how glycolysis and the Krebs cycle were. It's a little bit more all over the place, but hopefully by drawing it out, it will make a little bit more sense. But that is next time.